Okay, our next and final speaker in this session is Ronan Fleming from Leiden University, who's going to talk about multi-omics data integration and modeling.
Thanks to the organisers for the invitation to speak. Today I am just going to talk to you about a method we've developed for integration of multi-omic data with metabolic models. The basic approach here is you develop a biochemical network model, and you can use that as a means to predict cellular function. But then the way to make that cell type specific or condition specific is to use omics data to specialize it to a specific scenario. XomicsToModel is a protocol paper. It's a Cobra Toolbox extension. It's a set of executable tutorials and also some real-life examples of generation of a dopaminergic neuronal metabolic model using this technique, and it's all open source and openly available to everybody via these links. It's a fairly complicated protocol. It took us 5 years or so to develop. I'm only going to touch on a couple of bits here today. So essentially the idea is you can combine the computational analysis with experimental data generation and mathematical modeling and integrate those things together in this XomicsToModel pipeline to generate cell type specific models. This example here is illustrating for the dopaminergic neurons, but you could do it with any other iPSC derived cell type or another cell type you can culture in vitro, or even you could use it for in vivo data from any human tissue. So at the start, you begin with a generic network reconstruction, and some of you might be familiar with Recon3D, which is what we started with, which is pretty much now fully anatomically resolved, or maybe 90% atomically resolved metabolic network reconstruction. We then extracted from that a generic model. So what's the difference between those two? The difference between the generic reconstruction and the generic model is that the model satisfies certain mathematical assumptions. So when you want to ensure that everything's stoichiometrically consistent, you get a subset. When you want to make sure that every reaction can carry a steady state flux, then you get another a greater or smaller subset again. And when you want to make sure that every flux can be thermodynamically consistent, you get a smaller flux set of reactions again. So this figure here just illustrates those subsets for different human metabolic network models. And in particular, I draw your attention to the comparison between Recon3 and Human 1, which just shows that it's not really any difference of 70-odd reactions or so. So generation of specific omics data then would follow a similar approach as what's been described this morning. But in particular, we were interested in the metabolite uptake and secretion rates of dopaminergic neurons in culture. And so together with Thomas and Amy and their team in Leiden, we applied some new and established protocols to measure the concentration of fresh and spent medium, and then the difference between those gave us the rate of uptake or secretion of the metabolite. This type of data, together with transcriptomics and a bunch of other literature-created data I can't go into, all integrate together inside this XomicsToModel pipeline. But we generated an ensemble of models, and the ensemble of models generated because you're not really sure what the right parameters are. You're not for building the model. So, for example, what's the right transcriptomic threshold or something like this? And you're not really sure exactly what the right cellular objective is to represent metabolism in dopaminergic neurons, because this is unknown. It's a big question in the field. So we have to try generating multiple different types of models with varying the parameters and objectives. And then see which ones fit the data the best. So in particular, it was important to be thermodynamically feasible subnetworks. And it's fairly complicated, the reason, but essentially you can imagine it like this. So there are many, many cycles, like this one illustrated, with the red dots are the metabolites and the black lines connecting them are reactions. So there's many of these cycles that are omnipresent structural features of metabolism. But some metabolic network prediction algorithms will will predict flux going around in a cycle around these things, which is thermodynamically infeasible. So it would be equivalent in a river, if you have an island in a river, that there will be water flowing around the island, which you never see. So we wanted to make sure that we have a network that had none of these cycles in it. So basically, they're structurally there, but what can happen is you bounce. If you have a set of bounds that drive flux only in that direction, then this, and no matter what algorithm you use afterwards, you will always get cyclic flux. And so we wanted to avoid that. So that's the key algorithmic advance inside the XomicsToModel is that we can now, for the first time, generate these thermodynamically consistent metabolic networks. So we had this ensemble then, and the way we picked out of the ensemble about 400 models, which one were the best performing ones, was we took the uptake rates measured in Leiden and tried to predict the secretion rates. Or we took the secretion rates and tried to predict the uptake rates. And so the modeling approach and the parameters that best predicted or fitted, let's say, those data, that was the way we trained in a way the model to pick the best set of parameters and the best set of objectives. Then to validate it, we took separate data that wasn't used at all during the training phase from perturbed neurons, so uptake and secretion rates again. And so here we did leave-one-out cross-validation. So that the perturbation data was mitochondrial complex 1 inhibition and complex 5 inhibition, and we had the corresponding in vitro data generated as well ourselves in Leiden. And so this is showing you now the rate of uptake and secretion. The predictions are in red there. The diamonds, the blue with the error bars are what the measurements are. The diamonds are just showing that we can fit with all the data, and the red is the leave-one-out cross-validation. So what you see here in the end of the day is that we pretty good, qualitative accuracy, and most importantly, I think what we call moderate semi-quantitative accuracy. So you might not be impressed with a Spearman Rho for 0.48, but I can tell you in our field that beats anything, because most people in the constraint-based modeling field won't give you quantitative predictions. They will give you qualitative predictions. So there's still, of course, improvement to be made there, but I think it's a substantial advance. So especially since the model contains over 1,000 genes, over 2,000 reactions, there's 238 metabolites that are exchanged across the boundary, and we're only measuring a small subset of those illustrated by this overlap in the Venn diagram. So the model is largely under constraint, right? So then actually what was the most interesting thing for me in the whole study was what do we do when we are unconstrained or under constrained? How do we predict a unique solution when we don't have enough information to specify things uniquely? Well, that was down to the objective function, so the choice objective function. So I tried many different ones: 1 norm minimization, 2 norm minimization, 0 norm minimization, weighted versions of all of those. And we also, but the most predictive objective was maximization of entropy. So very fundamental reasons for this. Some of you who are information theorists will appreciate that. But essentially, maximizing the entropy is a way of making the least biased prediction given the data you have already. So in this case, we're maximizing the entropy of the unidirectional fluxes. So every reaction is split into a forward flux and a reverse flux. So how do we use this protocol or this pipeline then in practice? Well, we have multiple studies ongoing with that. This is just now one example. So a great PhD student working with me at the moment, she went through 74 clinical metabolomic studies of Parkinson's disease that have been published since the first one. And she found 928 metabolites reported to be associated with Parkinson's disease. Only 190 of those were replicated in more than one study. Out of those 190, 60 were reported to be exclusively increased, and 54 exclusively decreased, and the other ones were sometimes increased, sometimes decreased. So then you take those set of metabolites, then you can and feed them into the XomicsToModel pipeline. It can extract for you a subset of the generic model of human metabolism that it enriches for the pathways or the reactions, and then the corresponding pathways that are either associated with metabolites increasing or decreasing consistently in Parkinson's disease. So this is not like pathway enrichment, what we heard a bit about earlier today, because it comes ab initio from the stoichiometry and the network structure. It's not afterwards we add the labels, right? We show you these labels and the picture of which parts of metabolism they are. But the actual reactions and pathways that come out is ab initio. It comes out of the network structure. It doesn't come out of, it doesn't suffer from the bias of which label we put on the pathways or which label we put on the metabolites. So we were asked to maybe discuss open challenges, and so a little bit faster than I thought. So I'd like to do a little audience participation with you, if you would like. So what I'm not able to predict, and what I find the most, I suppose, hard thing to do in science is to predict which proposals that get submitted will get funded. So I'm going to introduce three rationales and three proposals that try to address those challenges. And then afterwards, I'm going to ask everyone to vote on which one of the three you think should be funded. And if you don't vote, then it means you're automatically wrong, because one of them did get funded, okay? So really, please join in and participate in this. So what's the problem with this approach that I just showed you? Well, the main problem with it, I would see, is that it's semi-automated. It took 5 years to do it. Now we have the pipeline that's quicker. We're generating draft models of macrophages and glial cells and things like this quickly. But the metabolomic side is, still takes a lot of time, and the modeling part is being partially speeded up. But the whole process, you don't just do the experiment and do the model. You typically have to go around in iterations. So the trouble with that is then you require all different disciplines to be together. You need all the PhD students to be around at the same time, or the postdocs to be around at the same time, no one to leave the project during the time, or you need 2 persons for every type of discipline. And then if someone leaves or gets a new job and then doesn't hand over to the next person, or 2 people don't get along, or whatever it is, it's really difficult to try and get this cycle to complete. And so I figure what we should do is we should instantiate this process in an automated system. And so this idea of this AutoMetabo proposal is to couple automated metabolomic analysis with automated model generation, so that the metabolic phenotype would be driven by the needs of the model. So we can use the models to identify which metabolites would be most important to measure in the next iteration to maximally reduce the space. This is an optimal experimental design. So then we could use that to pick the platforms to run. And the little robot would run through in Leiden and go to whichever mass spec would be most suitable, right? So it's not a way of replacing humans, really. It's about instantiating human knowledge into fixed processes, right? Because that's one of the big difficulties in multidisciplinary research. When someone with that key piece of knowledge leaves the group, you lose that. But if they put that into an automated system, you don't lose the information, because the robot won't forget. So that's AutoMetabo, if you like it. Another problem is that constraint-based models are growing in size. So I'm mainly speaking to you about metabolism, but these models have been generated for transcription, translation, and integration of transcription, translation with metabolism, etc. So they're going beyond the scope of metabolism now. But by the time you go beyond the scope of metabolism to, let's say, transcription, translation, the models get very large. And then if you have communities, like, for example, a microbiome, then you're going to get very large models, so in the order of hundreds of millions of variables. So we need a new framework for doing this type of modeling. But the existing constraint-based modeling software architecture is a bit like this. So you have different developers working in their pet language or whatever it is, and they generate their own internal framework, and then what's common to all the constraint-based modeling tools is that they call optimization solvers, and the best ones are industrial ones out there, at least the most accessible ones are the industrial solutions. So each one of these different software implementations implements their own interface to a whole set of solvers, right? And so I'm responsible for the Cobra 2 box, which is a MATLAB version, and so it's been around the longest. And what I find a little bit frustrating when people come along just implement it in different languages, why didn't they come and ask me what I thought was the main problems with the Cobra 2 box? I could have told them for free what to not do in the next time you're going to write it in another language. And so what I would instead do is something like this. So the idea is you'd have a hybrid relational database, so it's partially in memory and partially in storage, and there exists now already some basically they're like a cross between a database and an optimization solver. So you can write an SQL query, and then the model gets sent directly to an optimization solver, and the result comes straight back into the database. And the reason why I'd like it to be database-centric is all languages have their standard implementations of communication protocols with databases. So if you could agree on a standard database architecture to represent a constraint-based model, you could use this as a common tool that would allow people, let's say in MATLAB, to use the XomicsToModel pipeline, and then maybe in Python use an algorithm to make a prediction with that model, but without having to switch completely from one language to another, which is the practical barrier that stops people actually collaborating. You know? So if you like that project, that's high performance optimization for molecular systems biology. That's cost you $1.5 million. Budget is no limit, by the way, in this thing, but you get to decide. So another problem that I would say we have is that that study there, we did it on dopaminergic neurons, and a lot of work went into it, but we didn't find a cure for Parkinson's disease. I think we made some progress, but what you will find is that, certainly the Alzheimer's case is probably a perfect example, just like Parkinson's disease. They're so very complicated diseases, and it's probably not the same disease in each person, just like the asthma thing we heard before. So you're trying to build up a multi-omic picture of something nefarious, like it's not a single disease, there might be multiple different types of diseases, and you don't know how many. So which treatment for which type of disease or which subset of disease are you going for? And I'm thinking maybe what we should do is we should try to focus more on inherited metabolic diseases first, to understand more the disease process, because we know there at least the cause of disease, in most diagnosed cases, But even within, you can have individuals apparently that have the exact same genetic mutation, and they have two different phenotypes. So there's still complexity there, but at least you have an anchor to begin with, So the existing approach to diagnosis of inherited metabolic diseases is, you first go to a specialist. You probably don't get a diagnosis on the first instance because the doctor had maybe never seen that disease before. So they send you on to another specialist if they don't diagnose you incorrectly. And then that cycle goes around and around until they find the specialist that does the particular genetic or metabolomic test to diagnose that particular disease. And it's called a diagnostic odyssey. To some extent, whole exome sequencing has really sped this whole procedure up, but only in 50% of the cases can it be genetically diagnosed alone. So some other omics data or some other clinical information or whatever is going to be necessary on top of that. So in a way, this approach, this situation is improved, but it's not for the other 50%, let's say. So what we'd like to do in reconstruction, to use reconstruction and computational modeling of inherited metabolic diseases to accelerate diagnosis of inherited metabolic disease by working with patient registries of inherited metabolic disease patients, doing whole genome sequencing on them, and comprehensive metabolomic and proteomic analysis, then taking that data into standardized genomic analysis pipelines, modeling the enzyme structure using the likes of AlphaFold and things like this, and combine that also with the metabolic network modeling to so that we can try and direct the clinician in the first instance to a diagnostic test that might confirm quicker instead of going around in the cycle so much. So it's not meant to replace what you just saw. It's the current approach. It's just meant to try and avoid too many times going around in the cycle. Because the problem with the diagnostic tests is they target specific subsets of molecules or genes. But then you have to have been right with your prediction which one might be the causative gene. Otherwise, you just miss it. Sorry. So then you got 3 options. If you want option 1, put up your hand now. So maybe like 10. If you want option 2, put up your hand now. If you want option 3, put up your hand now. Okay, you're right. You're right. So that one was actually funded, and there's 20 odd positions open. So yeah, please get in contact if you're interested.